Today is the Braude lecture for Grand Rounds, and I'm just going to say a few words about Dr. Braude, who was a member of the Great Generation. He, after finishing just an internship, was drafted into the Army and spent most of the war, World War II, in Puerto Rico, which at the time was a kind of backward tropical island. And there he got a lot of experience with parasitology, which was just one of the many interests that he maintained through his life. So his connection to UCSD is that he was the first chief of infectious disease here, recruited by Gene Braunwald. At the time, the Bonner Plan was in existence. And so under the Bonner Plan, there were no preclinical departments. And so he was recruited to be both the chief of infectious disease and to be essentially the chair of microbiology, since there was no microbiology. And it was a just-in-time recruitment and appointment so that he could teach microbiology to the second-year medical students. Abe's had a firm belief that microbiology was intrinsic to the practice of infectious diseases, and he had been recruited from the University of Pittsburgh, where he ran the microbiology lab, and his only requirement to come here was that he be able to run the microbiology lab at UCSD. Avril Liebau, this is the Liebau Auditorium, was the chief of pathology here. And when Gene Braunwald told Avril that he wanted Abe and he wanted him to be the chief of micro, Avril said, no, only pathologists can run laboratories. And Braunwald said, make him a pathologist. And that's what he did. So Abe really believed that microbiology should inform the care and treatment of people with infections, and that from studying microbiology, we could learn a tremendous amount about the pathogenesis of infectious diseases. And that tradition has carried on, so that Charlie Davis, who was trained as an internist, took over the micro lab from Abe, and now Sharon Reed is running the micro lab, all ID trained physicians running the microbiology lab and making important contributions to patient care. So with that, I turn the microphone over to John Guatelli, who was also trained under Abe Braude.
True enough. Thank you, Josh. So I'm very happy today to introduce today's speaker, Dr. Kathleen Collins from the University of Michigan. Kathy earned her MD and PhD from Johns Hopkins University. She did her thesis research in the lab of Dr. Thomas Kelly studying DNA replication. From there, she went to the Brigham and Women's Hospital and affiliated institutions in Boston for training in internal medicine and infectious diseases. Kathy then trained as a postdoctoral researcher in the lab of Nobel laureate virologist Dr. David Baltimore at MIT. And in a groundbreaking Nature paper, she reported that the HIV accessory protein NEF, that's name is NEF, NEF, enables the virus to evade adaptive immunity, rendering infected cells resistant to killing by cytotoxic T lymphocytes. This was a seminal finding because it was the first example of an HIV gene with an immune evasion function, a paradigm that would subsequently be applied to the other so-called accessory genes of the virus. Kathy was then recruited to the University of Michigan, where she's now a professor of internal medicine, microbiology, and immunology. She delved into the cell biology of NEF activity and identified key cellular cofactors that NEF co-opted. She turned her attention to the HIV accessory protein VPR and identified activities of this protein that enable more efficient replication in macrophages, which is a potentially important cell type underlying HIV persistence. She also identified novel cell types, such as hematopoietic stem cells that can harbor replication competent viral genomes in treated patients and contribute to persistence despite our best therapy. And she's now developing NEF inhibitors towards the goal of sensitizing HIV to clearance by host immunity as part of a cure strategy. So in addition to running a cutting edge and robust basic research program in HIV virology and immunology with a huge list of trainees at all levels, Kathy has until recently been clinically active as an infectious disease consultant. She relinquished that role to become the director of the Medical Scientist Training Program and the Associate Dean for Physician Scientist Education and Training. She's the recipient of many awards. I won't list them. She's an elected member of the American Society of Clinical Investigation, the American Association of Physicians, the National Academy of Medicine. She's a member of the NIH Office of AIDS Research Advisory Committee and Early Stage Investigator Task Force. And she's the current editor-in-chief of JCI Insight. So how she does all these things, I don't know. But in summary, Kathy is just the sort of physician, basic scientist educator that I think Dr. Braude would have advocated for and did advocate for. So thank you for coming, Kathy. We're looking forward to talking the rest of your visit.
Well, I appreciate the history behind this lecture, which is so important. And thank you very much for the kind introduction. It's really fun to come here. It's been a long time for me. I think the last time I visited was late 1990s, to be honest. And it's just great to come back. So that's me. And thank you. Thank you to the UCSD Internal Medicine and Infectious Disease communities. It's a big honor for me to come here and to speak for the Braude Lecture and the Department of Medicine Grand Rounds. So I was asked as part of my talk to spend 3 to 5 minutes with no slides discussing something personally meaningful. And I think a lot of times when I talk to Department of Medicine groups, I like to reach out to the trainees who are often curious about, you know, how senior faculty came to where they are and you know, what did we do that worked and things like that. But in looking back, I'm always sort of struck by the fact that how nothing was really planned and how my career sort of grew organically from things that I discovered that I loved along the way. And I think being open to those things as you go along in your career and exploring is one of the best ways, because when you find out what you love to do, that's the thing that you're going to be successful at. And so along the way, one of the things that happened to me was the MD PhD path, because I discovered only late that I was interested in research. I was late in my undergraduate career when I got into a lab, and it struck such a chord with me that I ended up deferring my medical school admissions because I was stuck in the lab. I loved it. I didn't want to go. So, and then I discovered that there was actually a program that I could apply to during my year of, back then people really didn't take gap years. So I just call it my deferral year. And I applied and I was accepted MD PhD. And that was such an important path for me to really have the time post-undergrad to develop skills in basic science research and have that protected time to become a scientist. And it was critical to me to have that opportunity. And then as I was finishing my MD PhD training, I came out in the midst of the HIV pandemic and witnessed firsthand patients, young children, babies born to infected mothers who were dying or not even going home from the wards because the nursing staff expected them to die. There was nothing that you could do to help them. And I knew that I needed to work in HIV. And along the way, the mentors, of course, were critically important. I ended up in David Baltimore's lab after my internship and residency to study HIV. And it was work I did in his lab with other lab members and collaborators from MGH that really allowed me to be successful as a postdoctoral fellow, of course. And then the talented trainees who happen upon your doorstep, who come in and you don't know if they're going to be successful, and they don't know, and you give them the opportunity and they discover things you didn't expect them to discover. So I think just I would just encourage people to be open to things that you can become excited and passionate about, and take opportunities along the way as they make sense. And for young people, if you're interested in research, to really focus on research, and maybe later on take on some things that might distract you from research, which is what I've done recently. So in my later years, the dean's office reached out to me and said, hey, why don't you become an editor of a journal? And I said, editor of a journal? Why do I want to do that? But I did. And apparently I did, because now I am. And I love it. It actually was such an unexpected pleasure to be the editor-in-chief of the ASCII journal JCI Insight. I took it on after it was only a journal for 2 or 3 years. It didn't have an impact factor. And I built a board that was made up mostly of Michigan faculty, because that's what ASCII journals do. JCI is the other one. And now we have an impact factor of almost 10, I like to say. And pretty soon we'll be handing the journal back to ASCII, and they'll assign it to a new editor-in-chief. So if anybody here is interested in being editor-in-chief of JCI Insight for 5 years, think about it. And I'd recommend doing it as a more established faculty member, because it is pretty time consuming and it is hard to juggle with other things. Around the same time I took this on, however, I was also asked to consider being MSTP director, which I didn't know that I wanted to do either, but I love also being MSTP director. It's a bigger time commitment than being the editor-in-chief of a journal. But because the MD PhD program was so important to me as a young student, it was important to me to try to be a good MSTP director and to try to take up those reins and do a good job with that program. And along with that came an associate dean position, where I was asked to consider the role of graduate medical education and physician scientists, which I felt aligned with my job as director of MSTP, because all of our MSTP fellows graduate and they go on, of course, almost all to residency programs. And at least at the University of Michigan, there was no community for them of physician scientists. Once you became a resident, you were a resident. Once you were a fellow, you were mostly a clinical fellow. And these groups across clinical departments didn't connect the way they do in an MSTP. And so my goal was try to reach out across the clinical departments and bring physician scientists together in an MSTP-like community to share some career development activities, social activities, and benefit from physician scientist role models. So that's what I've started to do, which is, it's all been fun and challenging. And yes, it is very time consuming. And yes, it has been challenging to continue in my lab, but fortunately we have some data, which I will share with you. If I can advance my slide. Okay, there we go. So, so many of you, of course, don't need to be reminded of the HIV replication cycle, but because this is a grand rounds, I just want to make sure that everybody's on the same page. So I'll quickly walk you through it, because I'll be referring to many of these features throughout my talk. So HIV is a lentivirus, and it has a lipid coat in which are studied a relatively small number of envelope proteins. And the tropism of the virus is determined by the envelope protein, which interacts with the receptor CD4 and subsequently to a chemokine receptor CXCR4, CCR5, to gain entry to the cytoplasm, where the RNA genome undergoes reverse transcription to become a double-stranded DNA, make its way into the nucleus, and then become integrated into the host DNA genome. After which the virus uses the host transcription, splicing, and protein translation machinery to assemble the virus, which then buds from the plasma membrane and goes on to infect the next cell. I will, a little bit later in my talk, talk about another situation that can happen post-integration, which is for reasons that relate to a number of factors, including the cell state. At the point post-integration, the virus may become latent, in which case it may not transcribe or make protein as efficiently. And under that condition, the virus can live for the normal life of the cell until it becomes activated to make viral proteins, at which point the cell might experience some cytopathicity and die. Okay. So many of the genes encoded by HIV are structural, like GAG, POL, and envelope, and are required for the nuts and bolts of the virus replication cycle. Rev and TAT are needed for viral transcription and translation, but there are 4 proteins, VIF, VPR, VPU, and NEF, that aren't formally required for most of what it takes to make a virus protein. And these so-called accessory proteins are probably mostly important to disrupt the host immune response against HIV. HIV, of course, differs from coronavirus in many different ways, but even though coronavirus can make people extremely sick, people who survive the infection clear the infection by and large if they have a normal immune system, whereas people who acquire HIV do not. And there are many strategies of immune evasion, but today I'm going to focus on those that occur through the activities of NEF and VPR. We think that VPR is really important for counteracting components of the innate immune response against the virus, which is the first line of defense that the immune system has against the virus, and doesn't take any time to develop. It's immediately there. Whereas the adaptive immune response, of course, takes longer. It takes weeks to develop, but is equally important for controlling a virus. Okay. So, as I mentioned, I'm going to talk about the accessory proteins, VPR and NEF, and I'm going to talk about how VPR functions in macrophages to counteract the innate immune response through affecting transcription. And I'm going to talk about the role of NEF in adaptive immune evasion, which it accomplishes by down-modulating the host protein MHC class I from the cell surface to disrupt antigen presentation. And I'm going to describe the development of an inhibitor that we're working on that might help sensitize infected cells to the adaptive immune response if we can make it safer. And then finally, I'll talk about latency. And though the T cell target of HIV is the more abundant reservoir of latency, we have some good evidence that HSPCs can rarely be a latent reservoir as well and serve as a nidus of infection in optimally treated people. So the accessory protein VPR has been enigmatic. We still don't really understand what it does. But importantly, VPR is really important for infecting macrophages. And macrophages, like T cells and HSPCs, express the viral receptor CD4 and can become infected. And they're much less susceptible to the cytopathic effects of the virus. You can culture infected macrophages for weeks, whereas infected T cells die very rapidly. But one thing we and others have noticed that if you infect macrophages in vitro with a fully infectious and spreading wild-type HIV, you can get good infection fairly quickly, but if you mutate VPR with a frameshift mutation, the virus is definitely attenuated, and you can show that wild-type compared to VPR, wild-type and VPR mutants in many donors and see the same thing over and over again. This restriction that VPR overcomes can be overcome if you dump in a lot of VPR negative virus. So whatever is restricting growth, you can titrate out by just adding lots of virus. But especially at very low doses, spread of HIV in macrophages is highly dependent upon VPR. And this distinguishes itself from what you see in T cells. So these are activated CD4 positive T cells, and it's apparent that wild type HIV and mutant HIV that lacks VPR are very similar in the dynamics of virus production, although the time frame is much shorter. So over the years, we've done a lot of work studying exactly what it is in macrophages that VPR does to promote infection. And early on, we discovered that in the absence of VPR, there's a lack of HIV envelope protein, and that this restricts the ability of the virus to spread in macrophages. But if you mix in T cells after equal macrophage infection, now you can show that spread to T cells is also VPR dependent. And then lastly, we identified the mannose receptor, which restricts the expression of envelope by binding to mannose residues on HIV envelope. And I'll show you an example of that. So shown here is a Western blot analysis showing that in the absence of VPR, mannose receptor levels are high in infected cells as compared to uninfected, but wild type HIV suppresses mannose receptor expression, and that this corresponds, the suppression of mannose receptor expression corresponds to relatively high levels of envelope in the wild type, which is lacking in the VPR mutant. And in studies that I'm not going to show today, we've confirmed that if you knock down mannose receptor expression with siRNAs, that you reduce the requirement of VPR. And so one important role of VPR in the macrophage is to reduce mannose receptor levels. And we've confirmed that it's binding to envelope that's so important for mannose receptors restriction of HIV in macrophage, because we've mutated the mannose containing sites on envelope and shown that lacking mannose residues on the envelope, the virus also becomes much less dependent upon VPR. So the mannose receptor seems to be an important innate immune response that macrophages have to pathogens. Mannose is not normally expressed on host proteins, but the HIV protein envelope protein is so highly glycosylated that the mannose residues are not cleaved off as they should be in the Golgi. And this residual mannose on the envelope makes it susceptible to binding to innate immune response proteins like mannose receptor that are geared up to detect these patterns of pathogens such as mannose. So we tried to figure out exactly how VPR is targeting mannose receptor. And there's a canonical way that VPR targets host proteins in cells by linking them to a ubiquitin ligase complex, ubiquitinating the proteins and targeting them for proteasomal degradation. But we could never show that VPR was targeting mannose receptor via that pathway. And in fact, what we found was that VPR was reducing the transcription of mannose receptor. And so we thought, oh, I guess that means that unlike what people have thought about VPR in the past, maybe VPR's big effect in macrophages is that it alters the transcriptional landscape. So maybe in addition to mannose receptor, VPR is affecting other things within the cell. And so, of course, like everybody else, we decided to do single cell RNA sequencing. And it was important to do single cell and not bulk RNA sequencing, because HIV only infects a subset of cells in our cultures of macrophages. And you really don't want to sort infected macrophages to isolate the infected ones, especially if you're using infectious virus where there's not really a good way to sort them. And so we took primary PBMCs from healthy donors, isolated out the CD14 monocytic cells and differentiated them into macrophages in vitro, and infected them with wild-type HIV or a VPR mutant. We allowed that to spread in culture for 10 days and then did single-cell processing. And what we found was that in VPR-infected or VPR-expressing HIV-infected macrophages, there was a really dramatic down modulation of a lot of host proteins and a somewhat less profound increase in expression of HIV proteins in the presence of VPR. And when we looked at what was down modulated, we could see a lot of things that sort of made sense. So a lot of interferon-induced protein products were down modulated in the presence of VPR, like IFI6 is one dramatic one, ISG15, IFIT-M3, and mannose receptor is less dramatic, but it's there. And it's about in the same magnitude that we determined by qPCR. And so how do you then figure out, in the face of this sort of dramatic down modulation of a whole host of genes, how VPR is accomplishing it? Well, we used a software package called Homer, which takes known motifs of transcription factors and then computationally determines whether there are patterns of transcription factor motifs upstream of the genes that are differentially affected in your system. And using this approach, we found that the ETS family of transcription factors had motifs upstream of most of the genes. It was about 900 in total, either had ETS family transcription factor motifs or ETS IRF family transcription factor motifs, which is slightly different. And that this accounted for a big chunk of the genes that we found were affected by VPR. And then we looked, so here's another sort of table form of all the different motifs that we identified and the different transcription factors, and the commonalities amongst them was that they were, many different types of ETS family members. And then we looked for expression in our macrophages of these transcription factors, and PU1 was the transcription factor that was highly expressed in the macrophages that we were studying, and to a lesser extent, IRF6. And these two factors, these two transcription factors work together to regulate genes. And so the other thing, so I'll take a minute and talk to you about PU1 and IRF, how they work together. So IRF is induced with pathogen infection, and it's crucial for regulation of immune responses, especially those that are downstream of pathogen recognition, like downstream from toll-like receptors for people who know about those. And IRFs interact with transcription factors like PU1 that are expressed highly in hematopoietic cells, and especially macrophages for PU1. And the way that these two interact determines the specificity and magnitude of transcriptional events that are induced by pathogens. So this was something that we might expect that HIV would want to downmodulate. So that's very exciting. And the cool thing that we could do with single cell sequencing is that, I told you that this is a spreading infection. So there's some cells that are actually infected, and then there are some cells in the culture that have been exposed to the virus, but have remained uninfected. So computationally, we can find the cells and we can look at HIV proteins, and on our uninfected sample, there's no HIV proteins. We can find an infected subset in our wild type virus and bystander cells that have been exposed to wild type virus but aren't infected. And then in our VPR negative, we can find the infected cells, and we can also find bystander cells in the VPR mutant exposed sample. And generally, we had agreement, the same cells that expressed one HIV protein also expressed the other HIV protein, which is good. That's what we wanted to see. So, and then we could look and we could say, well, in the infected cells, how do they compare to the bystander cells, both in wild type and VPR mutant, and how does that compare to uninfection, sorry, the control that was uninfected. And for IFI6, which you saw was profoundly affected in the absence and presence of VPR, There's not a lot of expression in uninfected cells, as you would imagine, for an interferon-induced protein. In the presence of VPR, there's an upregulation, but it's substantially less than in the absence of VPR for the infected population. And interestingly, in the bystander cells, there was an upregulation in the presence of VPR, but it was much less than what the bystanders were experiencing in the absence of VPR. So this was really cool because what it meant was that the effects of VPR not only benefited the infected cell itself, but it affected the surrounding cells, making the surrounding cells, which of course are going to be future targets of VPR, more susceptible to the virus, and it would enhance spread in that manner. And we saw something similar with IFID-M3, although fewer cells express it, it seems. In the uninfected cells, we didn't see any. And similarly, in VPR infected and in VPR exposed, we didn't see IFID-M3 upregulated. But infected cells lacking VPR, you can see there's a substantial increase. And also in the bystander cells of the VPR negative exposed cells. And so these are just a couple of examples of this pattern, but we saw it sort of throughout many of the genes that are in this pattern where they're regulated by PU1 and IRFs and downmodulated by VPR. So how does VPR affect this change in its targets and in the bystander cells? Well, many people have contributed to our understanding of how VPR works, even though, as I said, it's enigmatic. We do know that it interacts with a ubiquitin ligase complex through the so-called VPR binding protein, which is also known as DCAF. And it's thought to bring host proteins that it wants to dispose of into this complex and then tag them with ubiquitin, which then causes them to be degraded. And in this way, the virus can manipulate the components of the cell in many ways, which VPR apparently does. And so we asked whether this was the case for PU1. So if VPR degraded PU1, then there'd be less PU1 around to transcribe genes, and that could be our explanation. And so we looked at PU1 expression either in the absence or in the presence of VPR. And you can see that there was less PU1 when we also expressed VPR. And we could restore the PU1 expression by treating with MG132, which is an inhibitor of the proteasome. And so this pattern fit our model of what we would expect VPR to do to a host cell target protein. In addition, we expected that VPR would physically interact with PU1 in order to bring it to this complex. And so the standard way of asking that question is to immunoprecipitate VPR or PU1 and then see whether the two come down together in the test tube. And in fact, when we immunoprecipitated PU1, we could detect VPR coming along with it in the complexes. And when VPR was present, we could detect a connection between PU1 and DCAF, the component of the ubiquitin ligase complex. We didn't see DCAF come down with PU1 in the absence of VPR. And when we mutated VPR so that it didn't interact with DCAF, we no longer saw an interaction either with VPR or with DCAF. And so this, again, fits the pattern of what we would expect for a host protein target of VPR. And then we started to get curious about something else. There's an enzyme called TET2, which alters the methylation state of cytosine residues and causes epigenetic changes in transcriptional regulation. And TET2 had previously been implicated as a target of VPR and was shown to be important for regulation of IFID-M3, which I told you is also a target of PU1 and affected in macrophages by VPR. And so we found a paper that said that TET2 interacts with PU1 and that together they can regulate genes. And so we asked whether we could detect it. And indeed, when we pulled down PU1, we could detect TET2. So we asked whether VPR might be targeting both PU1 and TET2 because they're all in the same complex together. And so again, we pulled on PU1 and saw that we could get all 4 proteins coming down together, which is supportive of the idea that they might all form the same complex. And this is how VPR can affect both pathways. So in sum, the way we think this is working is that VPR is targeting this transcription factor PU1, which probably interacts with other factors like IRF and TET2. And in this way, it down modulates the transcription of innate immune factors like mannose receptor and IFN-M3 and others. And that VPR does that by recruiting PU1 and associated factors like TET2 to a ubiquitin ligase complex, where these proteins are targeted for accelerated degradation through the proteasome. And in this way, VPR is able to rescue envelope protein expression in macrophages by disrupting the deleterious interactions between mannose receptor and envelope, and also affecting a whole host of other interferon-induced genes that also require PU1 for their transcription, and making the cell more compatible with the virus, and also making bystander cells in the culture more receptive to infection and promoting spread. Okay, so that's the innate immune response. But you also know that there's an adaptive immune response to infections. And that's really important for clearing infected cells because that's the way the body identifies an infected cell and kills it. And the way that cytotoxic T lymphocytes find and kill infected cells is that they have receptors that recognize host MHC class I protein, which presents peptide epitopes from the target cell. And in healthy conditions, these peptide epitopes represent the whole milieu of proteins that are expressed in a healthy cell. But in a tumor cell or in an infected cell, the peptides that are presented by MHC class I are aberrant. And CTLs are selected in the thymus in such a way that they will only recognize peptides that are aberrant. And when they're recognized, a program occurs in which the target cell, the cancer cell, infected with virus or bacteria, is eliminated. And that's the way the body gets rid of infected cells. So as John mentioned, when I was a postdoctoral fellow, fresh from my internship and infectious disease fellowship, I wandered into the Baltimore lab with a simple question, really naive and simple, actually, in retrospect, which was, why isn't HIV like flu? With flu, you clear the virus, you get sick, but antibody kicks in and immune cells kick in, and then the virus clears. Well, there's lots of reasons since then that I've learned that HIV evades the immune response, but I was focused on cytotoxic T lymphocytes because they're so important for getting rid of infected cells. And so I was also able to make a connection with Bruce Walker's lab at MGH because he had anti-HIV CTLs that he was amazing at growing these cells in culture and cloning them out and knowing exactly what peptide epitope they recognized and knew the affinity of the T cell receptors and everything about them. And so I got some anti-HIV CTLs from him, and I got some virus from the Baltimore lab, and they had these cool ways of tagging the virus so you could find it using flow cytometric approaches. And I used wild type HIV and some mutant HIVs that we had in the lab. And I just did an experiment really where we infected the cells, and then we stained them for MHC class I, and then asked what happened when you mixed in the anti-HIV cytotoxic T cells. And the viruses allowed us to pull out the infected subset from the uninfected subset and determine that infected cells have low MHC class I compared to uninfected cells. And when you add in these high affinity anti-HIV CTLs, you can't really get very much killing in a 4-hour in vitro assay. This nidus of infected cells remained. But when you did this same experiment with a virus in which there was a frame shift mutation that disrupted the NEF open reading frame, now the infected cells have normal MHC class I levels compared to uninfected cells. And when you add in the anti-HIV CTLs, you can see that you get a much more dramatic loss of this infected cell population. So at least NEF is one important component of the reason why HIV can evade the immune response. And this gave me a wonderful project to take with me as a starting faculty member to kind of figure out how did NEF do this? And over the years, through the work of some MSTP students and PhD students in my lab, and also, of course, the work of many other labs, including John Guatelli's lab, we were able to show that when you immunoprecipitated MHC class I proteins in HIV-infected cells, you also pulled down NEF, and that you could disrupt that by making mutations in the class I cytoplasmic tail. So we knew that NEF was interacting with the tail. And based on some work from a lot of people, we were suspicious that clathrin adapter proteins, which affect the trafficking of proteins inside cells, might be partners with NEF. And in fact, we're able to show that in NEF expressing cells, we could co-immunoprecipitate clathrin adapter protein 1 and NEF with MHC class I. And we're able to map the protein-protein interaction domains, which facilitated later studies by John Guatelli and colleagues that crystallized this three-way complex in a really lovely study that showed how these three proteins came together. And so that was very gratifying work that was a product of many contributions. But this complex formation that occurs probably in the trans-Golgi network disrupts the normal trafficking of MHC class I to the cell surface and instead sends MHC class I for lysosomal degradation. And a similar but very different pattern happens at the cell surface, where NEF targets the HIV receptor CD4 from the surface with a different clathrin adapter protein that John Guatelli has also studied, and targets it via this different pathway to the lysosome for degradation. So in this really cool way, NEF can do some very different things that end up looking the same from a big picture perspective, but involve really different and very distinct and unique molecular mechanisms. So since that time, we've been trying to determine whether we could find an inhibitor of NEF, thinking that if we could make the wild type HIV look like the mutant HIV that lacks NEF, then CTLs that were added or that developed in people would be more effective at clearing infected cells. And if we could give that to people, maybe we could get rid of these cellular reservoirs, not by itself, of course, but maybe with some other things. And so we screened, I don't know, hundreds of thousands of small molecule libraries using a high throughput assay that we developed, and didn't find anything that would reverse the effects of NEF. And so then we collaborated with David Sherman at University of Michigan, who he's, in addition to being a great chemist, he's also a scuba diver, and he goes out and he collects marine actinomycetes from coral and brings them back to the University of Michigan and grows them in the lab, and develops these secreted components of microorganisms and makes them available to University of Michigan community for screening. And so we screened about 50,000 of these products and eventually found a few that were very potent at inhibiting NEF. And we worked with them to fractionate those extracts and eventually came up with identification of glycomacrolides as a class of natural products that inhibited NEF. And these included things like bifillamycin and concanomycin, which are drugs, antibiotics that are known to work through inhibition of vacuolar ATPase, which is important for altering the pH of intracellular compartments, most notably the lysosome. And so we found that one of these, CMA, was a very, very potent NEF inhibitor at subnanomolar concentrations below which we were able to see lysosomal neutralization. And I'll just show you here the way, how potently it works. So here is the infected cell population in a NEF mutant. You can see there's high class I, as you would expect in the NEF mutant compared to the uninfected cells. In the presence of NEF, you get a down modulation of MHC class I, which is what makes them resistant to CTLs compared to the infected cells. When we treated cells with CMA, you can see that now the infected cell population resembles that of the NEF mutant. So we predicted, we hoped, that this was not just a reversal of MHC class I protein, but it also retained the HIV epitope so that anti-HIV CTLs could recognize and kill. And indeed, as we titrate in the CTLs with the NEF mutant, which is our positive control, we got loss of the HIV population. With the NEF, wild type NEF expressing, the effect of CTLs was greatly reduced. And when we treated with CMA and reverse class I down modulation, the cells are targets. So the class I that's on the surface is functional and it sensitizes the cells to killing. So this was very exciting. But how does CMA work? And we still don't really know how it works, but you can see that the concentration at which we get NEF inhibition is much lower than the concentration at which we inhibit the lysosome. So the canonical pathway whereby CMA works to inhibit lysosomal neutralization doesn't really explain what it's doing to inhibit NEF. So we don't know what the target is. So that's a major goal of the lab is what is the mechanism of action of CMA. And even though we have a nice window between the efficacy of the drug and the toxicity at 72 hours, it's probably not enough to actually treat people with this drug. And so we're trying to use this as a starting molecule, and we have probably 30 or 40 synthetic derivatives so far that have allowed us to crank up the difference between the effect of the inhibitor on NEF, which is our target effect, versus off-target effects, which we think are, for example, lysosomal neutralization and toxicity. Okay, so I talked to you about a couple different ways that HIV can evade the immune system through attacking the innate immune response and also the adaptive immune response. But we also know that there's another way that HIV evades immune response, and that's through latency. And we know latency exists because of this big experiment we did with anti-HIV drugs, where we treated people with amazing drugs that completely stopped the virus in its tracks. There was no more virus spread. And we were all so optimistic that these drugs had cured the disease. And then our hopes were dashed because we found out that as we stopped the drugs, the virus rebounds almost always. And that was depressing. But we learned a lot about the virus because what we found was that the cells that were remaining in the body were resistant because they weren't dying. HIV often is toxic and kills cells, but when the virus enters and causes a latent state, there's no viral proteins being made that are killing, or not enough viral proteins being made to kill the cells. And so if these cells are long-lived, or if they divide, they can propagate the viral genome for the life of the host. And so this latent reservoir is, as many of you know who are experts on all of this in this audience, that to cure infection, we need to think of ways to target this long-lived reservoir of latently infected cells. And so this is just a diagrammatic representation of what I told you earlier on, is that sometimes post-infection, because perhaps the state of the cell, or because perhaps of where the virus integrates, or perhaps some defect in the virus integrates and could be infectious, but it doesn't get turned on. It's silent. And so we need to work on this. We need to figure out how to get rid of these cells. So one important cell type that is able to support latent infection is the T cell, and that's definitely the most abundant reservoir. It's the easiest to identify. You can find this reservoir by sampling peripheral blood and activating cells, though they don't naturally make the virus in their latent state. If you activate the T cell receptor in a whole host of ways, you can stimulate virus relief and release and collect it and sequence it and show, oh yeah, there's definitely virus there. But my lab also found that another CD4 positive cell harbors HIV. And in contrast to T cells that preferentially take up the virus when they're activated and preferentially support an active infection, when we take HSPCs and infect them in the lab, HIV establishes a latent state preferentially. And then we can treat them with drugs like PMA and ionomycin or cytokines like TNF and get the virus to turn on. But it's challenging to work with this reservoir because CD4 expression on stem cells is quite low. So there's a restriction at the cell surface to infection. And so they're infected at a low rate. And also you have to get bone marrow to get enough cells to study the in vivo reservoir. But this rare infection of stem cells can be demonstrated in patients. Using highly purified stem cells, we've been able to amplify out HIV genomes from patients. And interestingly shown that these sequences shown in pink that we found in progenitor cells can also be found as a clonally expanded population in the peripheral blood and in bone marrow cells. And we can even find it secreted into plasma virus. And how do we know that it actually came from the same progenitor cell type? Well, some defective viruses are actually incredibly informative because, for example, in this case, there's a small deletion at the 5' LTR, not enough to prevent release of the virus in the plasma, but sufficient to make the virus replication defective. So the only way that this virus can propagate is through replication of the cells, division of the cells, because the virus is not infectious. It can't make virus particles and have the virus go on and infect. And in addition, we were able to isolate the integration site because this was a very predominant clone in this individual. And we were able to get the integration site and show that it was the same. And this is one example where we've been able to find the virus clonally expanded in cells that are probably targets of HIV, but we've also had examples of finding defective viral genomes that are proliferated and expanded from progenitor cells into CD4 negative cells that normally wouldn't be infected with HIV. And so while this is rare, it seems to be reproducibly identified, and we can find infected HSPCs at a low rate in about half the people that we study. And then since then, we've been expanding our studies into latency in hematopoietic progenitor cells using a latency probe. Other people have developed these, but this is the one that we developed. We put mCherry as a fusion protein with GAG, and then we have GFP expressed off a constitutively active promoter, or we think it's constitutively active. We have two different kinds of promoters that we use. And this allows us to use GFP to find all of the integrations and then to measure mCherry when the virus is in an activated state. And we can infect HSPCs with this virus and then sort out latent infection and then do single cell RNA-seq. And of course, the progenitor experiment is a really good one for single cell RNA-seq because when you culture progenitor cells, even though they're initially 99%, CD133, CD34 positive, so they're progenitor cells, as you culture them, they spontaneously differentiate down different pathways. They don't become a T cell or B cell or a macrophage, but they start to express some proteins that are not normally expressed by stem cells, but are expressed by more differentiated cells. And so you can begin to separate these progenitor cells into different groups based on their gene expression pathways. And you can look either in green at the latently infected cells or in red at those cells that have turned on HIV gene expression. And then compare that to other markers, like in purple here, we have markers of stem cells. And one thing that was cool about this experiment was it confirmed some of our prior cells, showing that latency is favored in these primitive cells that express CD133, but then upon initiation of differentiation down different pathways, you can see that viral infection gets turned on. And so with differentiation, you start to see the effects of the virus infection, but the virus stays quiescent and latent within this stem cell compartment. And this was a really cool figure made by my collaborator, Josh Welch, who's a computational biologist. And what he did was he took the RNA expression by our cultured stem cells and overlaid them onto a database of transcriptomic analysis from bone marrow, and was able to overlay our infected cells with the transcription patterns of uninfected HSPCs, and also showed how some of them were differentiating along the pathway that would be expected if they were able to differentiate in vitro, which they can't, into T cells because there's no thymus, but that they're along that pathway. And then we saw some that looked like they were, starting to go along and become monocytes, at least part of the way down that pathway. So it's nice to see that the transcriptional programs that we were seeing in our in vitro culture correlated with freshly isolated cells and what they were expressing. So in the future, what we're working on right now is using reporters and constructs in HSPCs to study the chromatin landscape of latent versus active infection. And we're studying some differentially expressed genes we've identified to see whether they're causally involved in either latent or active infection, or potentially could be biomarkers of latent or active infection. And the work that I showed today was a product of so many people's efforts. But I'm going to talk mainly about Maria Virgilio because she's still in the lab and she's still working, and she's done most of the work, the new work on VPR, and has also spearheaded a lot of the single cell sequencing along with the Welch lab. Chen Li and Joshua Welch were critical for all of the single cell transcriptomic work that I showed. And in HSPCs, I'll mention Val Terry, who comes from UCSD and worked here for many years. And she's really been the driving force with our patient studies on stem cells. And Mark Painter, who's currently a postdoc at UPenn, and Marianne Yapel have really moved the NEF inhibitor project forward. And so that is the end of my talk. I might have a couple minutes for questions before I know you all have to go. So thank you all.
Great talk, Kathy.
Thank you.
Thank you very much. So much work. So interesting. What do we have questions from the audience here? If there is, there's a mic. If we do, we have a microphone right here. And then let me go ahead and read a question from the chat, and then I'll get to some questions here. Don Rubin is asking, my favorite proteins have been ADAM10 and ADAM17 that interact with NEF and potentially regulate virus integrations. Have you looked at the effect of these metalloprotease expression in your system?
Yeah, that's a very interesting question. And metalloproteases have come up from time to time. So far, we haven't gone down that path. But clearly they have their effects on so many things that I wouldn't be surprised. But we haven't studied that in particular.
Anything else on the chat here? Okay. Well, can I ask what? Go ahead.
Yeah, oh, wow, sorry. I was curious if your dual reporter virus expresses VPR, does it?
We have different versions, and we have both plus and minus VPR forms of the reporter virus.
Yeah, I was interested because P1 is, it's a critical lineage determining transcription factor for many cells in the monocyte lineage. And so
Yes.
I'm curious if expression of VPR as you differentiate, restricts cells from differentiating down that lineage and preferentially pushes them towards other lineages.
You read our minds. We're trying to do those experiments right now. At the moment, we're trying to make sure that our reporter virus that we were thinking about using for this experiment expresses enough VPR compared to the parent virus. And it's definitely there. I just want to make sure that it's enough before we go down that path. But we do have several really nice, Maria in the lab has several different ways of turning our HSPCs into myeloid cells. And we can infect them post-differentiation, and we can take infected cells and then sort them and induce them to become macrophages. So that's kind of where we want to go. And we might predict that VPR would actually inhibit myeloid development and maybe development down other pathways too. And so that could influence what infected progenitor cells become. Yeah, absolutely.
So I'm not sure I understand what the mannose receptor is actually doing in the course of the infection. Is it binding virus so that it doesn't get out, or and thereby interfering with replication, or does it have some other fact?
It's a great question. So the question for everybody else is, I talked about the fact that VPR reduces mannose receptor, and that we think that this is important to counteract a restriction by mannose receptor. And it's a good question because I didn't have time to show that data, but we've learned a lot about what we think mannose receptor is doing. So mannose receptor has been studied forever. It binds to mannose residues, which are present on bacterial pathogens and also on some viral pathogens. And I told you that I touched briefly on the fact that HIV envelope protein has mannose. And mannose is initially added to most excreted proteins and most extracellular proteins in the ER, but then in the Golgi, mannosidase cleaves off the mannose so that host proteins don't normally have any residual mannose on them. But HIV envelope is so heavily glycosylated, there's this forest of glycans on the protein, and the enzyme can't get in to cleave off the mannose, so it's not processed. So HIV envelope leaves the cell with residual mannose on it. So that marks it in a way that tells mannose receptor and other mannose binding proteins that HIV envelope is foreign. And so mannose receptor interacts with the envelope protein and targets it for accelerated degradation in the lysosome. And if we cleave off the mannose residues for envelope, it's no longer susceptible to mannose receptor processing. But the tricky thing about some of those experiments is that we think HIV actually uses these interactions between mannose and mannose receptor to initially attach to cells. And so it's one of those things where it's a receptor that's helpful to the virus on the way in, but then the virus has to get rid of it so that it can get out, because mannose receptor envelope interactions inside the cell are deleterious. And in addition to binding up envelope protein that's free in the cell, mannose receptor is able to bind envelope on virions and target virions into lysosomes and reduce the amount of viral synapses that form between macrophages and T cells. I think it functions both intracellularly to bind envelope protein and also extracellularly to bind virions that are decorated with envelope protein.
Thank you all for coming. Thank you very much for coming.
Yeah, thank
Kathy.
you
It
for
was
those great questions. And thank you for inviting me.
All right.